share with you my views on this. And I hope that it will elicit some interesting discussion and responses on your part. I'll begin with a question. Here is a graph of our best understanding of life expectancy as a function of time. And if you look at the date, it goes back to the earliest of biblical times, and it runs up through the year 2000. And you see that there's quite a change. The average life expectancy, as far as we know, also in prehistoric times, was between 20 and 30 years. Now, obviously, some people lived a lot longer than that. Some people, unfortunately, lived a lot less. But the average, when you take into account all of the possibilities, was about 25, according to the historical record and from analysis of bones and skeletons, the prehistorical record. So something obviously happened about 1850 that caused a dramatic change in our circumstances that continues to the present day. What could that have been? Actually, it's the beginning of scientific research in medicine. Now, of course, the scientific method had been introduced, indeed formalized, several hundred years before, and had been applied first by Isaac Newton, then by the great German chemists, and the result of which was a deep understanding, but an evolving understanding, but already a scientific understanding of a considerable portion of physics and some of chemistry. But it wasn't really until the middle or late 1800s that we began to see the same sort of rigorous scientific approach applied to disease and to physiology. It wasn't until then that microbes had been identified. It wasn't until then that microbes were identified with disease. And so beginning in the late 1800s, and the names that stand out are, of course, Pasteur, Koch, Ehrlich, and others, we began to see an understanding of the causes of disease. And we also began to understand that very simple precautions, like Semmelweis's hand washing before exposure in a clinic, or later chlorination of water, proper sewage treatment. And then as immunology began to develop as a science, we began to understand that microbes could be combated in advance by vaccination. Of course, that had been known for hundreds of years in a non-quantified way, but it was studied in a quantitative way and made into a science. And so vaccine science developed in the early part of the 1900s. And you can see now, looking at the death rate, this happens to be a measure of death rate in the United States, a series of public health events which were put into place beginning in 1900. There's a spike in deaths, which some of you may recall was the last global pandemic that before the one that we recently experienced, the last major one, the influenza pandemic of 1917 to 1919 approximately. And then a very important event occurred in the late 1930s, which was a discovery of penicillin, which opened the era of antibiotics. And you see that the rate of death, primarily due to bacterial disease at that point, was reduced. And so we see over the course of perhaps 40 years, certainly 50 years, a virtual elimination. So unfortunately, it's not an elimination, but it's an extraordinary set of advances. First, related to viral diseases as a result of vaccines, and later as a result of antibiotics, reduction and control of, to a large degree, of bacterial disease. And so now, if you look at the overall rate of life expectancy statistics, what we've essentially done is eliminated the cause, or a major cause, of early childhood death. And as a result, you see a tremendous uptick in survival statistics. But of course, if you live longer, because as a result of not dying from infectious disease, you will eventually develop heart disease. So the heart and the cardiovascular system became the next major vulnerability. They were always there, but at a population level, they were not really revealed as much as when infectious disease was suppressed. And so I can well remember, and some of the older colleagues in the room probably remember, people in their parents' generation, my parents' generation, dying of heart attacks and strokes beginning in their 40s, frequently in their 50s, and typically in their 60s. And so life expectancy when I was born in 1950 might have been around 60 to 65 years for a man, and for women, perhaps 5 or so more years than that. And the major cause of this premature death, if I can call it that, were heart attacks and strokes, diseases of the vascular system. Well, let's look at those statistics. You see that starting from look at total cardiovascular disease on the top, as distinct from the breakdown into other forms of vascular disease. You'll see that actually the prevalence of, or the incidence, I guess, of cardiovascular disease is on the rise from about 1900 to about 1940. Well, I think you now understand why that is, because it was during that time that death due to infectious disease was gradually being suppressed. And when that battle was won, then the next major challenge, cardiovascular disease, started to take over. But look at what happened after about 1960. The incidence, the death rate due to cardiovascular disease has plunged dramatically, and it continues to plunge. And that's true in all categories, be it stroke, coronary heart disease, or chronic heart failure. So you might wonder what happened there. Okay, what enabled that? And so I'd like to tell you a little bit about that story in a bit of detail, because it illustrates the importance of basic science. And I could pick many, many examples. I could have picked many examples in infectious disease. And this lecture or talk could go on for probably several days worth of examples. But I picked this example because it happens to be something closer to the field in which I work. And so many of the participants over the years I have met, or I know people who knew them. And so this happened because of breakthroughs in basic science that gave us important classes of drugs. And there are today still two very important classes of drugs. Those of you who may be medical students or physicians obviously know about this, but the public knows about these very well too. The two major culprits in producing atherosclerosis are a combination of high blood pressure and high cholesterol. High cholesterol, especially in the form of low density lipoprotein cholesterol, or LDL cholesterol. And combating either one of these is a big help, but combating both of these together has added up to a decisive victory. That doesn't mean that we won't suffer from heart disease as individuals. Certainly we will, but our heart disease can be controlled, our vascular disease can be controlled, and its rate of progression can be controlled. And as a result, lifetime is extended, and people can live longer and higher quality lives. So blood pressure and cholesterol. And it's, of course, through drugs, and there are now many drugs that are available that provide various ways of controlling in various combinations these two major risk factors. So let's take cholesterol. It has a really, and there won't be time to talk about blood pressure, but let's talk about how we came to discover the drugs that control cholesterol. And those drugs, many of you may know, are called statins as a class, statins. And the first statin was approved for human use in the late 1980s, if I recall correctly. But the history of where the statins comes from or came from actually goes back to the early part of the 20th century. It's a work of basic science, of dozens of basic scientists, to say the least, over many, many decades, work that had an unpredictable course at the beginning and at almost every stage, as the best science always does. But let's go through the history of it. It actually starts with Heinrich Wieland and Adolf Windaus, who discovered the structure of cholesterol. Now, why were they interested in determining the chemical structure of cholesterol? By that, I mean its covalent bond arrangement of carbon, hydrogen, and oxygen in its covalent bonds, its structure as a chemical. If you don't understand the structure of the molecule as a chemical, you're certainly not going to have the basis for understanding what it does to the body, both in health and in disease. Well, even then, in the early 1920s, it was known from pathologists who looked at lesions in the blood vessels of people with coronary artery disease or other forms of vascular disease. There were clearly visible lesions called atherosclerotic lesions. They're very evident. If you've ever had or do have the chance to have one in your hands, as I did in medical school, you'll see that it has a fatty quality to it. And so it has fatty substances in it. And those fatty substances include the major culprit, cholesterol. And cholesterol could be isolated not only from these atherosclerotic lesions, but from other parts of the body. And it was characterized by Wieland and Windhaus, who discovered that cholesterol not only exists in isolation as a compound in these blood vessels, but is present throughout the membranes of cells. That's, of course, relates to its physiological role, but also bears in common many structural features, and indeed are metabolically related to other so-called sterols. Those include the bile acids, which we secrete into our digestive tract to act as detergents to help us consume foods, and other related structures. So the beginning of our understanding of how to combat cholesterol came from two German chemists who were really just following their curiosity and trying to ask, what was this chemical made of, and how did it relate to other chemicals that are found in the natural world, be they in plants or be they in animals such as us. The next major step came from a completely unrelated direction, a Swiss and a German chemist who were interested in the hormones that determine sex, which we call testosterone and estrogen. You might think, as a student, you might think, gee, haven't we always known about the sex hormones? Well, actually not. They had to be discovered. And these chemists were able to isolate from the blood and from primary sexual tissues of animals these substances, and they, to their astonishment, discovered that when they determined their chemical structure, they looked a lot like cholesterol. And actually, they looked a lot like the bile acids. And the answer for why that is that they all are formed metabolically. They are all synthesized in a common related metabolic pathway. And so this sort of illustrates another sort of important thing about basic science, which is you can pick almost any starting point, and if you get deep enough, you discover beautiful and profound relationships in every field of science in areas that you don't expect. So no one might have thought that the process of digestion would rely on very similar chemicals to the barrier property of a biological membrane to the determination of sex. But that was the case. And these scientists went even further. They found an even more fundamental relationship to the plant product rubber. Who would have thought that rubber would have anything to do with cholesterol in our blood vessels causing atherosclerosis? But indeed, it turns out that the so-called terpene unit of rubber is based on a repeat of 5 carbons in a certain pattern And then in cholesterol and in other sterols, you have 5 or 6 of these units of 5 that are put together. And some portions of that are then turned into a ring or multiple rings to form the steroid nucleus of cholesterol. I'm not going to bother you with the details of the metabolic pathway, but the core of the understanding, the kernel of the understanding that relates all of these chemicals to each other, is the fact that they come from this 5-carbon unit called isoprene. Okay, and I know that sounds very abstract and very chemical, but it turns out that when you crack the question of how do you make that 5-carbon unit, that's when you get statin drugs. So you see, looking ahead, perhaps another 30 or 40 years beyond this point, no one could possibly have known that these discoveries would lead directly, in the hands of others, of course, to the discovery of drugs that would control cholesterol and save lives around the world. But they did. The next major step was even more fundamental, because no one had any idea how a carbon-to-carbon bond was made in the body. And cholesterol, of course, has many carbon-to-carbon bonds. I think I just told you that the core of the synthesis of a steroid involves taking 5 carbons, I told you, a 5-carbon unit, and putting those 5-carbon units together, right? I think that's what I told you. But I didn't tell you how the carbons got together to form the 5-carbon unit, did I? Fritz Lippmann discovered that, and he discovered the very basic principle that enables the synthesis of carbon to carbon as well as other important bonds to make the various molecules, small molecules, as well as proteins and nucleic acids that comprise our body. And that involves the enzyme called coenzyme A. For those of you who may have studied biochemistry, you'll know that you will have heard of coenzyme A, because when you break down fatty acids, you break them down from long chain hydrocarbon to 2 units at a time that are attached to coenzyme A. It's like a 2-carbon unit, which is acetic acid or vinegar attached to coenzyme A. And likewise, when you synthesize carbon-to-carbon bonds, the high-energy activated acetate and related molecules that are linked to coenzyme A are grafted onto other carbon or other skeletons. And the energy that was stored in the thiol ester bond, as it's called, to coenzyme A, is utilized in this biosynthesis process. So again, on the face of it, this seems like a side story. It was actually quite possibly the most basic discovery made in all of biochemistry in the 20th century. But because of that fact, it provided an even broader illumination, okay, of a broader landscape. And that illustrates another important feature of basic science. Sometimes the answers to controlling disease come from truly unexpected places. No one would have thought that Fritz Lippmann's discovery of coenzyme A in a very odd strain of a bacteria, which turned out to be an auxotroph for phosphopantothenic acid, which turns out to be the key substituent of coenzyme A. No one would have thought that those very odd discoveries being carried out by this German scientist now working in the United States. I actually had the chance to meet him when he was old and I was young. He was completely incomprehensible. I once heard him give a lecture about coenzyme A, where he spent the entire time facing the blackboard and writing little notes in front of it that you couldn't read because he was standing in front of it. So here's someone who would not have survived in modern science, could not have written a research grant that would have been funded, at least in my country. But although in my country, we know that almost nobody's research grants get funded anymore. That's because of our political interference, which you hear about in the newspapers. But I won't go into that. So Lippmann made this monumental discovery that illuminated many, many things. And among the things that illuminated was how these 5 carbons get together. Next, in the 1950s and early 1960s, came Conrad Bloch and Fyodor Lunin. both German scientists, but now one, Konrad Bloch, working in the United States, and the other, Theodor Lunin, working in Munich. And they discovered the actual enzyme systems that synthesize cholesterol and also fatty acids, utilizing coenzyme A and acetate. So they discovered the exact details by which these molecules are put together. And now we're at the point where a drug can be discovered. Couldn't have been discovered before. It took all of this. And by the way, their ability, you might wonder why, are biochemists stupid? Why did it take so long to do this? Okay. And the answer is that the technology to be able to trace individual carbons to figure out how they got added came from here, it went to there, was only introduced during and after World War II as a result of the development of atomic energy. We had radioactive tracers. We had, unfortunately perhaps, excellent methods to enrich for deuterium because of its use in developing weaponry. And they were put to peaceful use. And with that came modern biochemistry, in which metabolic pathways could be efficiently discovered, and the enzymes that mediate each of the steps or each of these chemical transformations could be readily discovered. And that's what these scientists did. Now, as a result of that, they were able to identify each of the enzymes in the pathway that synthesize cholesterol. Okay. Now, I'm not going to show you that pathway. It has about 20 different steps. What Konrad Bloch and Theodor Lunin did was a monumental piece of biochemistry. When I was a professor at Stanford many years ago, we actually invited Konrad to come and give the lectures to the first-year medical students on how cholesterol is synthesized. And it's really such a beautiful pathway, but I don't remember staying awake myself through the entire lecture, even though he was a gifted lecturer. So it's a long pathway. I'm not going to subject you to it. And it was very important, but even then, it didn't pinpoint which of the many steps, 15 or 20 steps, in making that cholesterol molecule, which one would you want to poison with a drug? Because if you have a drug that prevents the synthesis of cholesterol, then actually the amount of cholesterol in your body will be reduced, right? And if the amount of cholesterol in your body is reduced, the amount that will end up in pathological plaques will also be reduced. Now, I know that that's a simple-minded statement, and it's not literally correct, but in fact, it is correct that lowering the right kind of cholesterol in the right way has the effect that I just described. But how did we learn that? Now we come to the modern era and the work of Michael Brown and Joseph Goldstein. And they discovered the answer to exactly that question. They studied human patients, people who had a rare genetic disease called familial hypercholesterolemia. And so they are two trained physician scientists who became interested in the genetic causes of disease. And there were families. This is a well-defined gene. The nature of the gene wasn't known. It's the protein it encodes wasn't known. The gene could not yet be mapped when they started working on this problem. But there clearly was a gene because you could trace the disease, the phenotype, from generation to generation in these families. And the affected individuals developed atherosclerosis as teenagers and generally died, if they were homozygous, in their early 20s of heart attacks and strokes. People who were heterozygous, much more common, of course, have longer lives, but develop premature atherosclerosis and die prematurely as well, but not nearly as dramatically. And what Brown and Goldstein did is they systematically discovered ways to correct these patients' cells in test tube systems. And they discovered in so doing that the key to the problem is the lipoprotein LDL. And they discovered that what was wrong with these patients is that they lacked an LDL receptor. Our body cells have specific receptors for low-density lipoproteins that clear them from the blood and allow the cholesterol and the fatty acids inside to be reutilized by the body so as to avoid making new cholesterol. So when your LDL receptor is taking cholesterol up from the blood effectively, it's delivering the cholesterol to the cells and throughout our bodies. And that LDL has two purposes inside the cell. Purpose number one, it provides cholesterol so we don't have to make it. Now, if we went on making cholesterol, even as we were eating cholesterol, that which ends up in LDL, if you eat cholesterol in your steak or what have you, or your eggs, it ends up being repackaged in your liver and it ends up in lipoproteins, ultimately LDL. Normally, if you eat a big meal with cholesterol, your body will make less, and therefore you maintain cholesterol balance. But if your LDL receptor is altered, in this case genetically, what will happen is you will accumulate the LDL in the blood coming from the food that you ate. You will not metabolize it, and it will cause pathology. Even worse, the signal in the body cell that the LDL that was taken up by the receptor provides, there's normally a signal that says to the cholesterol synthesis pathway, make less of these enzymes and shut down the pathway. Okay? And so your body not only accumulates the cholesterol you ate in the blood, it adds to the injury by making even more cholesterol. And that's why these people get sick. And that's what Brown and Goldstein discovered. And in so doing, they could pinpoint the signal that the LDL provides within the body cells. And it's that signal, in fact, shuts down a specific enzyme, one specific enzymatic step within this very complex pathway, okay? It's called HMG-CoA reductase. I'm not showing you the details of it. The enzyme has a name, and that is the target for statin drugs. So they discovered that if you could interfere with the function of this enzyme by adding a drug that directly inhibits it, taking advantage of the normal signaling pathway that controls the body cholesterol through LDL, but bypassing it all, then independent of whether you have this receptor, how much of it you have, how much LDL you have, that drug can control your body cholesterol. The next thing that happened is then that the drug was made. It was discovered by a man named Endo in Japan. And as a result of that is that Merck and company were able to do pivotal clinical trials. And in 1987, the first drug appeared. So let's just look back at what I've just told you. There's more than half a century of basic science that went into the discovery of these drugs that have totally changed the nature of our lives, okay? I take this drug. Many, if not most people my age take this or similar drugs. My family has a history of heart attacks. My grandfather, who you may have heard came from the town of Falecht, died of a heart attack in his 50s. He was about 50 years old. I might likely have suffered the same fate, but were it not for this drug. So I'm personally quite grateful that all this science had taken place. It began with the structure of cholesterol, an understanding of how cholesterol structure fits into the broader world of biology, and a world that has in common 5 carbon units, a detour to a very broad understanding of how carbon-to-carbon bonds are made in living systems, that enabled the next generation of scientists to figure out how the particular 5 carbon unit that I'm describing is made, and then the discovery of which enzyme within the synthesis of this 5 carbon unit was the point of critical regulation within the body that prevents the kind of pathology that I'm now describing. Then, with that in hand, large numbers of chemicals could be tested from various natural sources, and these chemicals would ultimately yield the statin drugs. So what are the lessons that we learned from this? I think there are two lessons that we learned from this. The first is that when Yuan starts sitting in the front row, your time is beginning to be limited. Is that correct? Correct. So I won't offer you the next half of my talk, okay? But I will summarize, I think, adequately about what we've learned about basic science. First, that the impact of basic science is broad, it's deep, and it's enduring. Each of the major steps that I outlined in this process of discovery was valuable in and of itself, had as valuable as it was for heart disease, added value equally for many other diseases and for a broader understanding of life processes and human physiology. So the value in many ways is hard to measure, but it's clearly very great. Where did these advances come from? Did they come from somebody in a government office who said, we need to have a 5-year plan for this, that, or the other disease? No, if they'd have done that, all the funds would have evaporated into thin air. A lot of papers would have been published, but none of this probably would have happened. It required instead individual scientists, individual creativity, intellectual freedom, the ability to make mistakes, because most of science is failure most of the time, and success is rare, and individuals who could follow their curiosity, independent of the specific outcomes that might have been anticipated. And as a result of that freedom, that lack of control, came an extraordinary combination of discoveries. And finally, a point that I made implicitly, but I would like to emphasize, science is international. It's completely unpredictable. It occurs across, it respects no borders. So these discoveries pinged and ponged from one part of the world to the next. Each scientific generation being aware of the work of the other through open publication, open sharing of information. And those are the lessons that I think we should learn about basic science. It's worth reiterating these because we are living in an era of organized, bigger science than we used to have. That invites itself to more government. And we need governments, obviously, to fund the science, but we don't want governments ultimately to control the science. But there's always that tendency because politicians and taxpayers rightly want to understand that their money is being well spent. And yet the kind of basic science that's the most impactful doesn't lend itself to measurements within even a 5-year frame that oftentimes are relevant to the true value that's being created. So we have a real ongoing challenge. We're seeing the challenge is becoming very clear in the United States today, in which the current administration is pushing back aggressively on fundamental research. But it's happening also in Europe. There's relatively less support for basic research within Europe, and it's more dominated by committees and sort of artificially constructed collaborations to promote international science and to spread money around, which I happen to think is a very good thing. Okay, but in balance, it's very important, but it can't get out of balance. And so I just think it's important that we all keep these things in mind. I think finally, in summarizing, the Academy of Science here in Moldova, by virtue of having meetings like this, by virtue of the exceptional leadership that the Academy enjoys, is benefiting from and contributing to global science by bringing together groups of people to discuss the important issues of the time. And for that, I applaud you, and I'm very happy to be a small contributor to that effort. So thank you.
Thank you very much, Professor Rothman, for this wonderful presentation. And I would like to note that we got 4 pages with questions from from students. Just a few. So the first question, you are a very active researcher, and the question is the following. What continues to drive your curiosity and passion for science after achieving so much?
Well, thank you, whoever asked it, for the compliment that I've achieved so much. To answer your very good question, I think what drives me still is the same thing that drove me all along. I mean, nothing has changed, and I am relentlessly curious. I continue to be ambitious, not for Nobel Prizes. People who get Nobel Prizes, if you ask them, they didn't do what they did because they were thinking at the time that they did it. Oh, it would be nice to go out and get a Nobel Prize. So I think I'll do this experiment today. That's not how science works. Science selects for a certain type of personality who wants to understand the world and wants to see beauty in it and would like to affect it. And so that's why I continue.
Thank you. As we are reaching the limit of our development at atomic scale of our chips and transistors, is there any possible reason where we will reach an age limit, even with all our advancements in medicine?
Okay. Well, there's sort of two questions embedded in that, sort of an interesting question. One is whether there's an ultimate limit to the age at which we can live. And the other, maybe more broadly, is there an ultimate limit to the progress we can make in medicine, irrespective of how long a person ultimately lives? Okay. There are two questions. I do think there is an age limit at some level. And what we're learning about aging more and more is that it's a disease of reactive oxygen species. I don't think anybody would say that's proven, but more and more, it seems as though chemical damage to DNA, proteins, membranes, and the like. And I don't see a way to ultimately control that because it comes from oxygen. Oxygen is the principal source of energy, along with light, light for plants, oxygen for us, that enables us to burn carbon to carbon bonds, combustion, and it's not 100% efficient. So the result of that is that there's always going to be oxygen products, and so I don't really see that. Now, as far as medicine goes, the broader question, I think it's important to recognize, as much as we've learned about medicine, man, we have so much more to learn. I mean, any topic in disease or physiology you get into, you find yourself in, as soon as you encounter a human body, as a doctor, you'll find yourself armed with some concepts, but a maze of possibilities and confusion and a shortage of specific ways to make diagnoses. So we have a huge amount to learn.
Yeah, thank you. There is a question here. How important is physics in medical research? In this connection, I would ask, how important is multidisciplinarity in your investigations?
Okay, so that's again a really good question. Johan, you have you keep coming up with good questions from the list, but I noticed that about 1 in 5 questions has a tick mark on it, so we're not going through the other ones. The physics has had a huge impact on biology over the years, not the least because of physical analytical methods. I described a moment ago how biochemically you can trace carbons in the synthesis of a molecule. Today, we use physics to image living cells at 1/10 or even a few percent, at 100 times the resolution that a light microscope allows, by playing tricks with light, super resolution imaging. There are so many ways in which physics impacts. More broadly, most discoveries that are really new come from the interfaces between scientific disciplines. Most of us are very good, hope to be very good practitioners to extend what we learn. And some of us are wanderers. And occasionally you get good practitioners from two different fields who happen to wander together and encounter each other. And you can find that the discoveries that are made generally come from those interfaces. Okay, my own training was in physics and chemistry, and I made major discoveries in cell biology that in previous generations, a field that was dominated by anatomists. The solutions came in my work from chemistry being applied. In the case of Randy Sheckman, who has visited this academy earlier and who I shared the Nobel Prize with, Randy's work involved primarily genetics, and he brought genetics to the discipline of cell biology, which was previously in the hands of George Pilati, the Romanian scientist that many of you have heard of. And George was actually trained as a classical anatomist and histologist. So in his interdisciplinary moment came when he moved to the Rockefeller University, and he interfaced with physics in the form of the electron microscope, which at that time was being used only for metallurgy. And at the same time interfaced with another lab that had developed and used the centrifuge in biology for biochemistry. And those were all, I think, essentially accidental encounters. So this is what we need. You need to promote as many accidental encounters as you can.
Thank you, dear Professor Rothman. I would like to note that you are an example for young generation, and we are happy that many young pupils, students are here. And we have here another about 45 questions. Remains for your next visit.
That sounds good to me.
Okay, thank you. Thank you. So, ladies, yeah. Da, vreau să menționez pentru cei care nu cunosc că domnul profesor Rothman a fost decorat cu medalia Dmitrie Cantemir pentru contribuția la succesul școlii de vară care a avut loc în luna iunie în sala Azorei Academiei de Știință a Moldovei. De fapt, școala de vară este o noțiune mai largă pentru că elevii vizitează și laboratoare pe parcursul școlii. Onorată asistență, continuăm implementarea agendei. Avem astăzi o prelegere publică despre noua neuroștiință și va fi prezentată de doamna profesor Joy Hirsch de la aceeași universitate, Yale University din Statele Unite. Profesor Joy Hirsch este o figură de referință în domeniul neuroștiințelor cognitive și sociale. Cercetările sale explorează modul în care creierul uman procesează interacțiunile sociale în viața de zi cu zi, combinând tehnici avansate de imagistică cerebrală cu modele psihologice sofisticate. Prin studiile sale, profesorul Hirsch a contribuit la înțelegerea modului în care oamenii interpretează emoțiile, intențiile și comportamentele celorlalți, oferind perspective esențiale pentru domenii precum educația, sănătatea mentală și interacțiunea socială în medii complexe. Pe lângă activitatea sa științifică, doamna profesor Hirsch este un mentor apreciat pentru tinerii cercetători, inspirându-i să îmbine rigoarea științifică cu creativitatea și să folosească știința pentru a înțelege mai profund natura umană. Prin munca sa, Joy Hirsch arată că știința nu este doar despre mecanisme și date, ci și despre înțelegerea profundă a interconexiunilor umane, contribuind la construirea unei societăți mai empatice și mai informate. So, dear Professor Hirsch, you have the floor here, please, and I wish you success.
Thank you very much. Thank you so much for that introduction, which I didn't understand a word of. So I have no idea what he said, First of all, I do want to thank our president, Ian Tiangiano, for organizing this whole week of events for us. It's been extraordinary and an incredible gift for both Jim and me to be here, to be participant in all of the things that are going on here in the National Academy of Science in Moldova. I have to say, following my husband is one tough act. He is a mighty speaker and his science is amazing. And following him is not something that anybody would want to do. But I'm looking forward to telling you about some new science, neuroscience. Now, instead of talking about the brain in terms of a single neuron, in terms of the micromolecular cellular biology, I want to talk to you today about the whole brain, the big brain, the live brain that's in your heads and how it actually relates to who you are, what you think, what you do, and how that squishy brain actually contributes to behavior. Now, let's start at this place with the human brain. The human brain has been a wonder in science for a very long time. It has been divided into many, many different classifications. And in fact, this is how we studied the brain early on. And this is a representative picture from Rothman, 1909, who discovered that the brain is actually composed of 52 cytoarchitectural regions. That is, there's different cell types throughout the entire brain. And one assumes that these different cell types do different things. But the discovery was just the different cell types. Now, for those of you who aren't neuroscientists, just let me orient you. We have the top of the brain, the bottom of the brain. Well, here, actually, yeah, let's get to the anatomy in a minute. The back of the brain over here, the occipital lobe, and this way over here, the frontal lobe. The temporal lobe is that structure down below. So we have temporal lobe, parietal lobe, occipital lobe, frontal lobe. And that's the structure of the brain. So we divide the brain not only by cytoarchitecture, but we divide it by its structure. We look at the anatomy of the folds, the tops and the bottoms, and it's known with extraordinary precision what each of those anatomical features are. So cytoarchitecture and anatomy. But we also divide the brain in terms of what those little parts do. Now, we call this the real estate principle. And by this cover of this magazine, this slightly cartoonish picture, I want to convey to you that we don't understand with great precision what the parts of the brain do. And in fact, that really is the main goal of much of neuroscience today, is to understand what is it that these parts of the brain do and how in the world they do it. And so this is the primary goal of modern neuroscience today. And unlike much of the science that Jim has told you about, this is a relatively new science. And so any of you out there in medical school or graduate school who want a new science that have many problems that are not solved, this is your topic. So hang in there, because there's a lot of stuff to learn. Now, we have learned a great deal from modern technology, particularly magnetic resonance imaging. Now, how many of you guys have been in a scanner before? You know about MRIs, you go in the scanner, you look at the anatomy, your legs, your feet, your legs, your lungs, whatever. You can also tune that scanner so that it can give you information about how your brain is working. So as illustrated here in this picture, you put somebody in the scanner and you tell them to hold real still, and then you have them do a task. And if your scanner is working properly, you can look at the images from that scanner and determine what parts of the brain were doing what parts of your function. So this technique called functional MRI absolutely revolutionized neuroscience. And can you imagine? One could look into the normal human brain and understand what parts of the brain were doing what things. This was absolutely amazing. And in fact, in many of my lectures, I've referred to this as the real estate principle. Specific things in the brain, specific real estate does specific things. So now let's start with another whole fact to start another site, a different topic. Now, what functions, if you could study the human brain and you wanted to know what parts of the brain did what things, what functions would you consider most important? Well, the early neuroscientists considered that the basic sensory functions were important. We learned about how vision works, how sensory and motor systems works, how thinking works, how cognition works, how executive functions work. But we put somebody in the scanner and we can't ask them to do tasks that are interactive with other people. Now, as it turns out, human beings are profoundly social. Profoundly social. That means, as human beings, what is our most favorite behavior? We interact with other people. We do it all day long, from the first day we are born to the last day of our lives. We interact with other people. Well, that interaction, those social systems, cannot be studied in a scanner. We put individuals in the scanner, we ask them to do things, but they're all alone in that tube of the MRI scanner. So we've learned a great deal about things that might relate to social systems, but we don't know very much about social systems from the scanner. But let's look at the first thing that implies a social system, and that is face processing. So if you're in a scanner and you want to study social processing, you can't really study the real thing. So what do? You show people pictures of faces, because we know that faces are very, very, very important to human social interactions. So in order to do that, what we do is we put somebody in the scanner and we show them intermittent pictures of faces and objects, faces and objects, faces and objects. And a lot of people do this for about a decade. And what we learn is really very important. We learn that the brain has devoted an incredible amount of real estate to understanding the processing of faces. It says to us that faces in all aspects are extremely important to the human brain. Okay, we test this hypothesis out by studying using electrophysiology with monkeys. And sure enough, the same thing, we understand that this whole temporal lobe, the whole temporal ventral aspect of the brain is related to the processing of faces. Well, that tells you something pretty important, that faces are pretty important to humans, and they have to do with our social interactions with each other. Okay, but there's a huge knowledge gap, because what we've learned in the scanner relates to single individuals. But what we really want to understand is how the brain works in live interpersonal interactions. We can't do that in the scanner. So what in the world do we do? Well, the first thing we do is think about what we need to have. What is there about a social interaction that we need to understand? Well, in a situation like this, we have multiple things happening. We have somebody that's sending information to another person using his or her face, her voice. That information is received. That information then is processed and sent back using voice and face, language information is received again. And there is this dyadic cycle of coordinated social cues that include facial expressions, productive speech, that have to do with this behavior of social interaction that we would like to understand. How in the world are we going to understand this? So that's just the introduction to my talk. So the roadmap, what I'm going to talk about, is the paradigm shift. That is the new technology that we need to understand live and interactive social interaction from the point of view of what's going on in the brain. I'm going to present some very new evidence for this live processing and how it compares to the static face. And the argument is that live interactions are very important, and the brain treats them very differently than anything that you can present to the brain in a static and simulated mode. I'm going to, as a scientist, we like models. I'm going to present to you a model of the brain in live and in live interaction conditions having to do with face-to-face processing and eye-to-eye contact. And then I'm going to end by testing this model in looking at the neural correlates of eye-to-eye contact in autism. And we'll probably stay on time. So here we go. So the paradigm shift. We can't study in live interactions in an MRI scanner. And so we have to develop a new technology, which has been developed. It is a technology using optical images that puts electrodes and optodes on the heads of individuals so that we can image brain activity under live and interactive conditions. And so it looks like this on the heads of the individuals. The detectors are head mounted. They're tolerant of movement, so people can be moving around and naturally interactive during the experimental conditions. Temporal resolution is amazing. We're imaging at about 30 hertz as opposed to functional MRI, which is about 1.5 seconds. Spatial resolution is quite terrible, but good enough. It's about 3 centimeters. We say here it's the spacing of the optodes on the head. The underlying anatomy is not acquired. It's assumed from a general template. But one of the incredible advantages of doing this type of imaging without a high magnetic field, what we have in the MRI, is that we can add on many other behavioral measurements. So we can image, in addition to the imaging of the signals from the brain, the gathering of the signals from the brain, we can gather eye movements at the same time. We can image the face. We can look at behavioral measures, physiological measures. And so we have a suite of measurements that combine not only the brain signals, but also the behavior that's associated with them. And it turns out that that's an incredible advantage. I'm not going to spend a lot of time on the details of how it works, but I'm assuming that all of you in science and medicine want to know a little bit about this. This is not magic, that there's a real signal. The signal is acquired because, first of all, the signal comes from the brain. And the signal that comes from the brain is a signal that is induced by the fact that single patches of neurons that are doing the job actually are metabolically active. And any metabolically active patch of neurons recruits highly oxygenated blood. So there's this recruitment process. The part of the brain that's active is recruiting blood to that area. That blood is highly oxygenated, which is a signal that that is the area that's active. So what we do is in this case, for optical image, we look for the signals that are associated with the task that are highly oxygenated. And we can do this by emitting wave light into the brain. So those little optodes that you saw on the heads of the picture of the participant have emitters. They're shooting a very low power wavelet of light into the brain, and it is diffused and detected. We know what goes in, we know what goes out, and we can measure what happens underneath based in terms of the amount of blood. Now, this is what we call a proxy. We're not actually measuring the neurons, but we're measuring where the neurons are active. So that is our signal. This is what it looks like. So here we can see 15 seconds is about how long it takes for a task to be observed. So supposing that you have someone that's doing something simple like a finger thumb tapping task, just a simple task like this. Well, this is activating my right hand, left side, left motor strip. And if I have an optode right here on the left side, right next to that motor strip, when I'm doing this, the signal will go up. And then I stop doing the task, it goes down. I start doing the task again, it goes up and down and up and down. And that's a perfect indicator of the task that is going on in that local area. So that's an illustration of the signal. All right. This is not a new technique. It was discovered 1977, but it was only in 1993 that it was discovered by Britton Chance that we could actually measure neural activity in the brain, as I've just described. This technique was not widely used until we started using it for what we call hyperscanning, that is the imaging of two people simultaneously. If you want to image one person and learn a task about single individuals, for heaven's sakes, put them in the scanner because it's a lot easier to do. But if you want to image two people simultaneously and you want to understand what's going on between them, then this is what we need to do. And so here's a basic sort of picture of what it looks like in the laboratory. Two people are looking at each other. We have a glass screen in front of between people. The glass screen is what we call a smart glass, and we can run a current through it, and it's light. You can see through it, or if we run a current through it, it becomes dark, so we could time perfectly when people are looking at each other and when they're not. So in the blue bars here in this timeline below, people can see each other usually for 3 seconds, and then it's off, and then it's on, and then it's off, and then people rest for 3 seconds. And that's a general paradigm for functional studies of these kind. Okay. So the first thing we want to know is, does it really matter that the brain is looking at another live person? Does it matter? And so we did studies where we have people just looking at real faces, like you saw, versus a video face. Or we can have people looking at real faces, as illustrated, or with just a simple robot face. And the answer is, look at the brain here, those highlighted patches of the brain. That is the right-hand hemisphere, and that's the supramarginal gyrus. And that is a very, very clear signal that yes, it matters a lot that you are looking at a real person as opposed to a simulated person. So that the data are very clear and very strong that the brain has devoted specific real estate for processing live faces. So we can begin to then say, okay, well, this old model from the scanner of the ventral stream looking at faces has to be enhanced with another module, that module that says, okay, parts of the brain are specific for live faces. But why? Why should that be so? What in the world about a live face is different from a simulated face? Well, one hypothesis is that this right supramarginal gyrus, this big patch you saw that's specific for live faces, might be just processing something about a spontaneous facial expression. Well, okay, so we can study facial expressions. We know that the face makes many kinds of expressions. People use AI and they can divide the face up into many different configurations. Those configurations can be detected very rapidly, and the amplitude of the signal can be calculated. And we can actually use that as a signal, as a regressor, or as a correlate to the neural processes. So, okay, so we can ask what part of the brain is actually interested in these faces. Well, as it turns out, in this experimental paradigm, we have somebody, a face watcher, watching someone that's designated to express a expression based on what he or she sees with a movie. We take those signals, as I said, use them as a correlate on the neural responses of the face watcher, and these are the results. Sure enough, that this supramarginal gyrus cares a huge amount, as you can see by that big cluster there, a huge amount about facial expressions. So that's pretty exciting. But now, the supramarginal gyrus might also be interested in some other aspects of social interaction, like what about eye-to-eye contact? You think that supramarginal gyrus is interested in eye-to-eye contact? Well, we test this hypothesis by actually measuring eye-to-eye contact between two people. And this is during the time that they're looking at each other. We know how much time they're actually looking in each other's eyes. And we can use that as a correlate on brain responses and ask the question, does the supramarginal gyrus activity actually correlate with the times of people are actually looking at each other? And the answer is absolutely, without question, no. This is one failed hypothesis that the supramarginal gyrus could care less about eye-to-eye contact. And you can see what's happening. The top of the brain cares a lot about eye-to-eye contact, and it's very specific. Well, the first thing this means is that we actually have to change our model. So our model, we start with our brain again. This is the right side because most all of these social systems are right-sided. We start with the ventral system, the static faces, the live faces, and now indeed the interactive faces, the faces associated with eye-to-eye contact. And here's a new model. Well, now this model suggests that these two new systems that we've just added to the brain model might actually be different from each other, from the static systems by electrophysiological activity. Anyway, we can find other ways that differentiate these two higher level, more dorsal parts of the brain during live face-to-face and eye-to-eye contact. And so we asked a very specific question about these two systems. Are they associated? Are they different from the other systems by their electrophysiological signals? We would expect that they would be. And so this is another test of hypothesis. And this is one of the things that you will all know as you go into science, is that there's no test of any one hypothesis is ever really sufficient. We keep testing the hypotheses over and over and over again until we actually make them fail. But this one is one that was reasonably successful. In this case, we ask about the electrophysiological activity in the case of these high-level regions when we're looking at the face of a robot. And you can see that in the first part of the time series, that high amplitude indicates that, yes, a face is being noted by those areas, but there's not much happening afterwards. But in the case of the live face, those two areas are very different and very active. And so we consider this also a successful hypothesis that these two high-level areas of the brain associated with live faces and live social interaction, as indicated by eye-to-eye contact, are indeed distinguished by the electrophysiology as well as the neural responses. Okay, so we have a new hypothesis, and we ask, why does this matter to us? Does this actually give us any insight into any of the conditions that we might be interested in a clinical situation? And of course, autism is one of the first ones that comes to mind. And the reason for this is that we know a lot about autism spectrum disorder, that the reason that we are interested in here, I'm going to get back to this, is that it is particularly characterized by difficulties in social communication and interaction, and including reluctance to make eye-to-eye contact. It's one of the primary defining characteristics of autism. And so in particular, we would expect that our patients with autism would, based on this new model of hierarchical visual processing, might indeed be affected specifically in the dorsal aspects of the brain, specifically with respect to eye-to-eye contact. And now, because of these basic science experiments that I just showed you, we actually are able to test this hypothesis. And so I'm going to show you in the remaining time that we have just a few studies that test this hypothesis with patients with autism. And so, as I said before, the model predicts atypical functions of the neural correlates related to eye-to-eye contact, particularly in the dorsal visual stream, patients with autism. So here, this is the eye-to-eye contact data that I just showed you with typicals showing that eye-to-eye contacts are correlated with activity in the dorsal stream in the right hemisphere. And now, if we do this with our autistic individuals, consistent with our hypothesis, I want you to note that there is a zero activity in the dorsal stream and almost all of the activity associated with eye-to-eye contact. By the way, our patients made eye-to-eye contact perfectly well. If you put an individual, these are high-functioning individuals, ask them to go in the scanner or in your experimental paradigm, and you have them make eye contact with a partner, they can do it perfectly well. But in this case, all of our autistic participants were using an alternative neural circuitry. An alternative neural circuitry. We refer to this as a ventral stream to process the same information. When we analyze the data in a much more rigorous way, we see exactly the same thing. Dorsal stream for typical, ventral stream for autistic individuals. A very powerful finding, actually, in looking at the neural correlates of what's going on. Now, does this have anything to do with the symptomatology? And as a clinician, you might ask, well, so what? What does this tell me about what I know? What do I know about my patients? So first, let me tell you that we as clinicians measure symptomatology in autistic spectrum disorder using a scale called the ADOS score, and it's the Autism Diagnostic Observation Schedule. Autism is diagnosed behaviorally, so you watch the child interact with other people. And so we have an ADOS score on all of our diagnostic individuals, and we can ask how this ADOS score actually is affected by the measures of eye-to-eye contact. The first thing that we did was use AI. And we dumped all of our data into one big package. And we said, all right, computer, if you are so smart, tell us which one of our patients are autistic and which ones of our patients are typical. And it turns out our machine can do this about 76% of the time. Now, this is very, very good. And AI, these AI guys, they're not so smart. You guys think AI is so smart? Forget it. It's not. 76%. Most clinicians could do it much better than that. But anyway, our machine does it 76%. But that's not what we were really interested in. We said, okay, this is good documentation that our brains of our autistic individuals are different from the brains of the typical individuals, but does this have anything to do with the symptomatology? And so we took the output from the AI machine. In order to classify whether somebody was typical or autistic, you have to spit out a number, and that number tells you how certain the machine is that the person is autistic or not. We call this the support vector machine output. That is the y-axis on that graph there, the ADOS scores, which the machine did not train on at all. So this was a really high test of the AI. It did not train on the ADOS scores. And you can see that correlation is very, very good. It's 0.72. It says that in the case of live eye-to-eye contact, symptomatology really is predicted by this eye-to-eye contact. And it's not true when the individuals are looking at a video. Okay. I can go on. I think in the interest of time, I'm going to skip a couple slides. The slides that I'm going to skip simply say that what we can learn from this technique is that this dorsal stream associated with eye-to-eye contact and symptomatology is atypical, that we have a great deal more to learn about the specific neural circuitry in autism and in normal typical interactions in general. So let me just whip past this, some numbers. We as scientists like numbers, but let me summarize just in the interest of time. Okay. So first thing, I've summarized the autism studies that I just told you about, that the live two-person FNIRS and eye tracking data confirm the behavioral predictions that neural correlates for eye contact differ from individuals in ASD and those that are typical, that the eye contact activity in the dorsal stream for typical, and it's in the ventral stream for the autistic. Aha, see. Okay, let's skip this one. Okay. All righty, guys. Just stand there. Just stand there a minute. I got a couple more things to say. These findings they support this model of hierarchical visual processing. And I just want to point out, making kind of a point of what Jim was making, that there's a whole lot of basic science that stands behind our ability to think about this, our clinical cases in a rigorous way. And this is my final wrap up. I want to impress that this is a new science, and that I hope I've inspired you to think about the new science of the living, naturally behaving brain as a new exciting area that you might consider going into. This is just a small step to a huge new landscape that's ahead of all of us as scientists and clinicians. That the active brain, the real live active brain, is an extraordinarily exciting territory for understanding complex behaviors. And I think I've illustrated some groundbreaking methods. Hope I've sort of broken some barriers that we can't study the brain in its live and native form. That it's possible to apply this clinically, and I hope in the end that I've inspired some of you to think about some of these incredible new directions for a new pathway in social neuroscience. And before my last word is that no work is ever done alone. And my work is done by many people, many students, many people who have come with me on this journey. And for them, I owe a great deal. So thank you.
Yes, thank you. Yeah, dear Professor Joy Hirsch, thank you very much for the very interesting, wonderful lecture. We actually planned lectures from Professor Rothman, and there were also a lecture from Professor presented by Professor Professor Hiroshi Amano. But this was actually very, how to say, unexpected, but on the other hand, we appreciate very much that you proposed to give this lecture. And there was a decision of the Presidium of the Academy of Sciences of Moldova. Let me to announce. So Nicolae Emilescu Spătaru Medal is awarded to Professor Joy Hirsch, Yale University, United States of America, for outstanding contribution in neurosciences and collaboration with the academic community of the Republic of Moldova. Decision of the Presidium of the Academy of Sciences of Moldova from October 2nd, 2025. So congratulations. From here, because you should, yeah.
Down in the hard fabric,
Yeah, you see, so actually, your husband is,
it's easier on his.
Okay,
Then
okay.
it's a hard fabric.
yeah.
Thank you so much. This
Please.
completely unexpected. Thank you. I appreciate your audience and the opportunity to share this work with you. I hope it had some inspiring aspect of it.
Yeah, thank you so much. We wish you to continue your scientific activity, you and Professor Rothman. And of course, we expect that you will come back to Moldova because, first of all, Moldova is a nice country. Second, since Professor Rothman has roots here, of course, we should make use of this and to create sustainable bridges, scientific bridges, and I would like to say bridges of good interaction between people. That's as important as the scientific bridges. And you actually, your talk actually was very interesting even from this point of view. Well, ladies and gentlemen, do not leave, nu plecați, că o să facem o fotografie de familie. Deoarece două prelegeri și a fost, într-adevăr, domnul profesor, so would you maybe you say just a few words, but we will invite you to make a lecture in the near future, but now just a few words, So, thank you. O să facem o fotografie toți împreună. Înainte de aceasta, domnul profesor, please say a few words. Just
I'd like to thank the president of the Academy of Sciences. It's really fantastic to have so much young people in the audience. First, sorry, I am Manuel Prieto from Lisbon University in Portugal and past president of the International Union in Biophysics. And just to emphasize 3 points, you had really 2 fantastic lessons from these professors, absolutely amazing. Just allow me to emphasize 3 topics raised by Professor Rothman. Science is international. Basic science is absolutely essential. Even in my country, in this moment, we have problems fighting for basic science. This is something new. 3, most of you were very young during the recent COVID pandemic, but science is at danger. You remember the mad things about anti-vaccination movements and stupid things that went around. So please fight for science. Science also, it's totally interdisciplinary. Myself in biophysics, I came from physical chemistry. I use lasers and equations to study proteins aggregating. This is Alzheimer and ion pumps. Potassium ions going through membrane channels. But it's basic science. Well, it has relevance, for instance, for epilepsy and some other things. What else? So basic science, interdisciplinary, science is international, fight for all of it. Science is not also, I have friends in all the worlds. Science is not compatible with racism and xenophobia, which is coming also in Europe all over, as you know. Well, you are young people, the future in Moldova, it's in you, and that's really what I can say. I'll be available, I'll be around to talk with you. It will be my pleasure. And thanks, Professor Jan, really.
Thank you very much. Thank you. Onorată asistență, vreau să vă atrag atenția că ne urmăresc online mulți, spunem așa, multe persoane care sunt aproape de știință și apreciază foarte mult munca oamenilor de știință. Și iată domnul profesor Prieto din Portugalia, avem aici domnul Ioan Pavel, este primar din Dumbrăveni. Vă rog să vă ridicați în picioare, care colaborează cu cercetătorii din Republica Moldova. Eu vă spun, colaborează într-o modalitate care este, eu consider, foarte bună, fiindcă ne susține știți, noi în cercetare uneori avem nevoie de o susținere și adesea ea e financiară pentru a mai realiza unele proiecte și domnul primar de Dumbrăveni, ca și multe alte persoane, chiar aici joacă un rol foarte important și noi aducem mulțumiri. Acum, eu vă rog pe toți, I would like to ask you to approach the first lines și să vă aranjați în așa mod ca să fim aici o semilună, așa. Și ați văzut luna când a fost sângerie? Da, o semilună și o să facem poze. So we will make a family photo and you please, a family photo, you should come here. Da. Tudor, te rog să vii încoace în față și să aranjăm așa ca toți să se vadă. So,
Would you like a?
yeah, please come. And you.
Nice to meet you. What's it? Okay.
Tudor, ia bine, să facă și